Welcome to Last Week in Science. I'm your host, William Logian, and today I'm going over CRISPR. What is it, how does it work, where is it useful, and is it safe? Find out after this break. CRISPR, short for Clustered Regulatory Interspace Short Palindromic Repeats, is a revolutionary technology that has transformed the field of genetic engineering. Discovered in the early 2000s, CRISPR is part of a natural defense mechanism used by bacteria to protect against viral infections. Scientists have harnessed the system to create a powerful tool for precisely editing genes in a wide variety of organisms, including humans. At the heart of the CRISPR system is an enzyme called Cas9. Cas9 acts like molecular scissors, capable of cutting DNA at specific locations. The process begins with the design of a short RNA sequence that matches the target DNA sequence researchers wish to modify. This RNA, known as guide RNA or gRNA, directs Cas9 to the exact spot in the genome where a cut is desired. Once Cas9 makes the cut, the cell's natural repair mechanisms kick in. Scientists can then harness these repair processes to introduce specific changes to the DNA sequence, such as inserting, deleting, or replacing genetic material. The simplicity and precision of CRISPR-Cas9 have opened up numerous possibilities across various fields. In medicine, CRISPR holds the promise of treating genetic disorders by correcting mutations at their source. For example, researchers are exploring the use of CRISPR to treat diseases like cystic fibrosis, sickle cell anemia, and muscular dystrophy. In agriculture, CRISPR can be used to create crops that are more resistant to pests, diseases, and environmental stresses, potentially improving food security and sustainability. However, the rapid advancement and widespread potential of CRISPR have also sparked significant controversies and ethical debates. One of the primary concerns is the risk of off-target effects, where the CRISPR system may inadvertently cut DNA at unintended sites, potentially causing harmful mutations. While researchers are developing methods to increase the accuracy and specificity of CRISPR, the possibility of unintended consequences remains a critical issue. Another major controversy surrounds the use of CRISPR in human germline editing. Germline cells are those that give rise to eggs and sperm, meaning that any changes made to these eggs can be passed on to future generations. In 2018, Chinese scientist He Jiankui announced that he had used CRISPR to edit the genomes of twin girls to make them resistant to HIV. This announcement was met with widespread disapproval from the scientific community and raised significant ethical concerns. Critics argued that the experiment was premature, lacked proper oversight, and potentially exposed the children to unknown risks. The incident highlighted the need for strict regulatory frameworks and ethical guidelines to govern the use of CRISPR technology in humans. Ethical considerations also extend to the broader implications of CRISPR on society. There is a concern that gene editing could lead to a new form of eugenics, where individuals with desirable traits are selectively bred, potentially exacerbating social inequalities. The possibility of creating designer babies with enhanced physical and cognitive abilities raises profound questions about the nature of human identity and the potential for genetic discrimination. Furthermore, the use of CRISPR in agriculture and the environment possesses its own set of ethical and ecological challenges. While genetically modified organisms created using CRISPR could help address global food security, there are concerns about the long-term effects on biodiversity and ecosystem stability. The introduction of CRISPR-edited organisms into the wild could have unforeseen consequences, and rigorous risk assessments are necessary to ensure environmental safety. To address these ethical and social concerns, it is crucial to involve diverse stakeholders in the conversation about CRISPR technology. This includes not only scientists and ethicists, but also policymakers, industry leaders, and the public. Public engagement and transparent dialogue are essential to building trust and ensuring that the development and application of CRISPR are guided by shared values and responsible practices. In addition to ethical considerations, there are practical challenges that must be overcome to fully realize the potential of CRISPR. One of the key technical hurdles is developing the CRISPR components to the target cells efficiently and safely. Various delivery methods are being explored, including viral vectors, nanoparticles, and direct injection, each with its own set of advantages and limitations. Overcoming these delivery challenges is critical for translating CRISPR-based therapies from the lab to the clinic. Another area of active research is the development of next-generation CRISPR systems. Scientists are exploring new variants of Cas enzymes with different properties, such as improved specificity, reduced immune responses, and the ability to target a broader range of DNA sequences. Innovations in CRISPR technology continue to expand the toolkit available to researchers, enabling more sophisticated and precise genetic modifications. CRISPR is a groundbreaking technology with the potential to revolutionize medicine, agriculture, and many other fields. Its ability to edit genes with unprecedented precision offers new hope for treating genetic disorders, enhancing crop resilience, and addressing other pressing challenges. However, the rapid development of CRISPR also raises significant ethical, social, and technical questions that must be carefully navigated. As we move forward, it is essential to balance the promise of CRISPR with a commitment to responsible innovation, ensuring that the benefits of this powerful technology are realized in a way that is ethical, equitable, and safe. That's all for this episode of Last Week in Science. I hope you enjoyed, and even more so, I hope you learned something. I'll see you the next time that science happens.